Welcome to our journey into animal diversity. Today, we're going to explore one of the most fascinating and vital processes in biology, osmoregulation in fishes. Imagine living your entire life submerged in water, yet constantly fighting the threat of either drying out completely or swelling up like a balloon. This is the daily reality for every fish on Earth. By the end of this lesson, you will understand exactly how these incredible animals maintain their internal balance and why a saltwater fish cannot simply survive in a freshwater lake. Let us dive right in. To understand how fishes survive, we first need to understand the physical forces acting upon them. This brings us to the concept of osmosis. Why does this matter? Because every living cell requires a very specific balance of water and salts to function properly, generate energy, and send nerve signals. Osmosis is the natural movement of water molecules across a selectively permeable membrane, always flowing from an area of lower solute concentration to an area of higher solute concentration. Think of a tea bag in hot water, but here the water moves while the trapped salts cannot. For a fish, its delicate skin and gills act exactly like that tea bag, constantly exposing its internal fluids to the relentless pull of the surrounding ocean or river. Now that we understand osmosis, let us look at the invisible force it creates. This force is known as osmotic pressure. Conceptually, osmotic pressure is the exact amount of physical pressure that must be applied to stop water from moving across the membrane completely. If a fish does not actively fight against this pressure, its cells will either swell up and burst or shrivel up and die. The continuous, energy-demanding process of fighting this pressure to keep internal salt and water levels perfectly stable is called osmoregulation. It is the ultimate biological balancing act, and it dictates everything about where a fish can live and how its body must be structured. This invisible pull of water we just discussed can actually be measured. We use the Van 't Hoff equation to calculate the exact physical force exerted by different aquatic environments. Conceptually, this formula shows how physics and biology intersect, revealing the exact pressure a fish is fighting against. Let us break down the symbols. Pi represents the osmotic pressure, usually measured in atmospheres. The variable I is the Van 't Hoff factor, telling us how many particles the salt breaks into. C stands for the molar concentration, or how salty the water is. R is the ideal gas constant, and T is the absolute temperature in Kelvin. Physically, if the water gets saltier, meaning C increases, or warmer, meaning T increases, the osmotic pressure pulling on the fish's gills increases dramatically. Before we move on, let us see this formula in action with a concrete example. We want to calculate the osmotic pressure of typical seawater at a warm temperature of 298 Kelvin. We are given a salt concentration of 0.6 moles per liter. Because sodium chloride splits into two ions, our Van 't Hoff factor is 2. We chose this formula to reveal the immense invisible force pushing against a marine fish. We multiply the number of particles, the concentration, the gas constant, 0.0821, and the temperature. The result is 29.36 atmospheres. Physically, this means the ocean exerts a crushing pressure equivalent to 30 cars pressing down on a small area, constantly trying to suck water out of the fish. Having calculated that massive osmotic pressure, let us explore how marine fishes actually survive it. Imagine being stranded in a scorching desert, surrounded by water you cannot drink. That is the daily reality for marine fishes. They are classified as hypoosmotic regulators. This means their internal body fluids have a much lower osmotic pressure, or saltiness, than the surrounding seawater. Because water always naturally flows toward the saltier side to dilute it, the ocean is constantly sucking vital water out of the fish, primarily through its delicate gills. If a marine fish does absolutely nothing, it will literally dry out and turn into a living mummy while swimming in the middle of the ocean. To combat this severe dehydration we just described, marine fishes have evolved incredible biological machinery. First, to replace the constant water loss, they must actively drink large amounts of seawater. However, drinking seawater brings in a massive toxic load of excess salt that could easily poison them. To get rid of this salt, they utilize specialized structures in their gills called chloride cells, which actively pump the salt back out into the ocean. Furthermore, their kidneys are highly adapted to conserve as much water as possible, producing very small amounts of highly concentrated urine. Without these specialized cells and water-conserving kidneys, marine life as we know it simply could not exist in our salty oceans. Now that we know how they survive, let us quantify the exact severity of the dehydration threat. We can model the rate at which a marine fish loses water using a simplified version of Fick's law of diffusion. Conceptually, this equation helps us predict how fast water escapes based on the physical properties of the fish and the water. The variable J represents the net flux, or rate of water loss. P is the permeability constant, showing how easily water passes through the gills. A is the total surface area of the gills. C out and C in are the salt concentrations outside and inside the fish. Physically, if the gill surface area A increases, or the difference in saltiness increases, the fish loses water much faster. The negative sign simply indicates water is leaving the body. Let us apply this water flux equation to a specific marine fish to see its daily struggle. We are given a gill surface area of 50 square centimeters and a permeability constant of 0.02. The internal salt concentration is 0.3 osmoles, while the harsh ocean is 1.0 osmoles. We use this formula to find the exact volume of water escaping. First, we subtract the internal concentration from the external to find the osmotic gradient of 0.7. This is the invisible engine driving water out. We then multiply this gradient by the area and permeability. The final answer is negative 0.7 milliliters per hour. Physically, this confirms the fish is losing nearly a full milliliter of vital water every single hour, forcing it to constantly drink and pump out salt. This diagram beautifully summarizes everything we just calculated about marine fishes. Look at the blue arrows pointing out of the gills. This represents the passive water loss due to osmosis, driven by the high salt concentration of the seawater. To compensate, you can see the blue arrow at the mouth showing the fish actively drinking seawater. Now, follow the red arrows pointing out of the gills. These represent the specialized chloride cells actively pumping the toxic excess salt back into the ocean. Finally, at the bottom, the small yellow drop illustrates the scanty, highly concentrated urine produced by the kidneys to conserve water. This entire system is a continuous, energy-demanding loop keeping the fish alive. Now, let us flip the scenario completely and look at freshwater environments. Imagine sitting in a wooden boat with hundreds of tiny leaks. Water is constantly seeping in, and if you do not bail it out, you will sink. This is the exact situation for freshwater fishes. They are known as hyperosmotic regulators, meaning their internal body fluids have a much higher osmotic pressure than the pure river or lake water around them. Because water naturally flows toward the saltier side, the fresh water is constantly flooding into the fish's body through its gills and skin. If the fish does not actively fight this inward flood, it will swell up like a water balloon and its cells will literally burst. To combat this constant flooding of water into their bodies, freshwater fishes use a completely different set of survival tools. First, they almost never drink water intentionally, as they already have too much of it. Instead, their highly efficient kidneys work overtime, acting like a biological water pump to produce massive amounts of very dilute, watery urine. They are constantly bailing out the excess water. However, peeing so much causes them to lose valuable salts that are hard to replace in a freshwater environment. To fix this, specialized cells in their gills actively absorb scarce salt from the surrounding fresh water, pulling it into their bloodstream against the concentration gradient. This allows species like trout and bass to thrive. To understand the immense workload placed on a freshwater fish's kidneys, we can calculate the exact rate at which it must produce urine. Conceptually, because the fish must keep its internal volume perfectly stable, the water coming in must exactly equal the urine going out. In this formula, U represents the required urine production rate in milliliters per day. The symbol K is the water permeability constant, indicating how leaky the skin and gills are. A is the exposed surface area. C in and C out represent the salt concentrations inside the fish and outside in the river. Physically, if the internal concentration C in increases, the difference in saltiness grows, meaning water floods in faster, and the required urine output, U, must dramatically increase to prevent swelling. Let us look at a freshwater trout living in a mountain lake to calculate its daily water intake. We are given a permeability constant of 0.5 and a total surface area of 100 square centimeters. The internal salt concentration is 0.3 osmoles, while the pure lake water is just 0.01 osmoles. We use this formula to find exactly how much water is flooding in. We subtract the lake's low concentration from the high internal concentration to find a net osmotic gradient of 0.29. Multiplying this by the area and permeability gives us 50 times 0.29. The final answer is 14.5 milliliters per day. Physically, this means the trout's kidneys must produce and expel exactly this much dilute urine every single day just to stay alive. This diagram perfectly illustrates the freshwater survival strategy we just calculated. Notice the blue arrows pointing into the fish. This is the passive water gain flooding in due to the fish's salty interior. At the mouth, you see a clear indication of no drinking, as the fish already has too much water. To handle the flood, look at the large yellow stream at the bottom, representing the copious amounts of dilute urine the kidneys must constantly produce. Finally, the red arrows pointing into the gills show the specialized cells actively scavenging and absorbing rare salts from the fresh water to replace what is lost in the urine. It is a beautiful biological adaptation. We have covered a tremendous amount of ground today, so let us summarize the most important ideas. First, osmosis is the fundamental physical force driving water toward higher salt concentrations, creating a relentless osmotic pressure that all fishes must fight. Second, marine fishes live in a dehydrating environment. They survive by drinking seawater, pumping out toxic salts, and conserving water. Finally, freshwater fishes face the opposite threat of constant flooding. They survive by acting as biological pumps, producing massive amounts of dilute urine while actively absorbing scarce salts. You now understand the incredible biological machinery that allows fishes to conquer the diverse aquatic habitats of our world.